name is Vijay Gupta and you are watching biology classes. So students, in this class of biology, today I am going to teach you a new topic, that is kingdom fungi. So in this video, I will tell you about the introduction, general characters and a special member of kingdom fungi, that is Amanita muscaria. So as we all know that currently I am making all my videos for my 11 students. I am just teaching you your chapter number 2, that is biological classification, in which today I'll tell you about the kingdom fungi. So let's start the video. First of all, about the introduction of this kingdom. The first term is mycology. It's a branch of biology which deals with the study of fungi. So you can say mycology is the study of fungi. You can say fungi in British English and fungi in American. So study of fungi is termed as mycology. Now the next term, there is a name of a scientist, that is P.A. Michaeli, and he was the father of mycology. Now general characters. So the first point is they are cosmopolitan in distribution, means fungi are found everywhere. So they are cosmopolitan in distribution. They may be in the air, on the land, in water, everywhere. So they are cosmopolitan in distribution. Now the next term is the body of a fungus is thread-like or tubular. The body may be thread-like or tubular. Now I would like to tell you with the help of an example, that is, it is the bread which we eat at our home daily. So it is a bread. Generally, we know that there is a fungus which is found on the bread, that is rhizopus. It is easily found in our home. So it is the rhizopus, a fungus also known as mucor. So in this fungus, the plant body is like this, means there are numerous thread-like structures which are attached together and to form a network. So this network of the thread is termed as mycelium. And a single unit of mycelium is termed as a hyphae. Hyphae is a thread which form a network-like structure, that is called as mycelium. So in the first point, the body is thread-like or tubular. So in rhizopus, the body is thread-like, and in some other fungus such as Amanita, the body is branched and tubular, tube-like structures. As you can say, there is an underground mycelium in Amanita. So these are the tubule-like structure. So the mycelium may be tubular or the thread-like. Now the next point is, in the plant body, thread-like hyphae are present. As I told you that in the plant body, the thread-like structures are present, which are known as hyphae, which forms a net-like structure known as mycelium, and these hyphae fuse together and to form a network-like structure that is termed as mycelium. Now next point is, the hyphae may be septate or aseptate. Now what is this? Suppose that it is the mycelium or it is the hyphae of a fungus, and again I am going to make another hyphae. So if the septa are present between the cells, if the septa are present in the hyphae which divide the cells, then this type of thallus is termed as septate. So these are the septa and these are the nuclei which are present inside the cell. So when the septa is present, the mycelium is known as a septate mycelium. So it is the septate. And when the nuclei are free and there is no septa, so then the thallus is known as aseptate. Simply, when the septa is present, when the septa are present between the cells, then it is septate, and when the septa are not present, then it is aseptate. So next point is, the cell wall is made up of chitin. As we all know that chitin is a type of carbohydrate. I have told you in my the video of arthropoda that the exoskeleton of arthropods is made up of chitin. So here again chitin is present in the cell wall. Suppose it is a single cell of a fungus and it is the outermost layer of the cell, that is the cell wall. So the cell wall of the fungus is made up of chitin, or you can also say fungal cellulose. So it is the chitinous cell wall. Next point is mostly achlorophyllous. Most of the fungus are achlorophyllous, means when the chlorophyll is absent, that the fungus is termed as achlorophyllous. And non-vascular means when the vascular bundles are not present in the fungus, there is no need of vascular bundles, so they are non-vascular. Vascular bundle means xylem and phloem. Two types of vascular tissues are present. So when the vascular tissues are absent, then it is termed as non-vascular. Now, in next point, I'll tell you about the mode of nutrition. So the mode of nutrition is parasitic, saprophytic, or symbiotic. There are three types of nutrition. First is parasitic. When the organism obtain their nutrition from the body of a living organism, then it is termed as a parasite organism. Most of the fungus are parasite. They may found on the body of human being or any other animal, and sometimes they are also found on the plant body, so they are the parasitic fungi. Next is saprophytic. When the organism or when the fungus grows or obtain the nutrition from the dead organic matter, then it is termed as a saprophytic fungus. And the best example is Amanita muscaria, as you can see in the diagram. So these are the dead organic matter through which it obtains its nutrition, so it's a saprophytic fungus. Now the last one is, that is symbiotic. Symbiotic means when two organisms live together and both are benefited each other, then this type of relationship is known as symbiosis, and the organisms are termed as symbiotic organism. The best example of symbiosis is lichen, where algae and fungus both have a symbiotic relationship and both are benefited by the activity of the other. So these are the symbiotic forms. Now I will tell you about the reproduction. There are three kinds of reproduction which is found in the fungus. The first one is vegetative, second asexual, and the third one is sexual. Now I will tell you one by one. So first of all, vegetative reproduction. There are several ways of vegetative reproduction which can be seen in the fungus. So there are maybe fragmentation, fission, and budding. So one by one I am telling you. First one is fragmentation. Fragmentation means when the plant body or when the thallus of a fungus may break into numerous fragments. Suppose that it is the thallus or it is the mycelium of a fungus, and it breaks into numerous small fragments. So these are the fragments, and each fragment is capable to produce a new hyphae or a new mycelium. Then this type of vegetative reproduction is termed as a vegetative reproduction. I repeat my sentence. When the plant body or when the thallus of a fungus breaks into small fragments and each fragment is capable to produce a new fungus, then this type of reproduction is termed as fragmentation, and it's a kind of vegetative reproduction. Now the second one is fission. What is the meaning of fission? The division into two equal parts. Suppose that there is a fungus named yeast. It's a unicellular fungus. When it is divided into two similar parts with the help of a cell wall, then this type of reproduction is termed as a fission or a binary fission. So it is the fission in which a single cell divided into two equal parts or a two daughter cells. So this type of vegetative reproduction is known as fission. Now the last one is budding. Now what is budding? Suppose that it is yeast and unicellular fungus. Some outgrowths are produced from the cell, and these outgrowths are known as buds. First of all, these buds grows, and after that they detach from the cell wall, from the cell body, and each bud is capable to make a new cell. So this type of reproduction is termed as budding. Means when the reproduction takes place with the help of buds, then it is known as budding. So there are three ways. Generally there are three ways. which are fragmentation, fission and budding, which are generally found in the fungus. So these are the vegetative reproduction methods. Now the next one is asexual, and again I'll tell you about three methods, which are zoospores, conidia and sporangiospores. So one by one. Number one, that is zoospore. Suppose that it is the hyphae, and these are the cells of a hyphae. Each cell produces a few special structures which are pear-shaped, uninucleate consisting of single nucleus, and biflagellate means consisting of two flagella. This special structure is termed as a zoospore, and this zoospores swims freely for some times with the help of their flagella, and after that it settles down and germinate and develop a new mycelium or a new hyphae of a fungus. So when the reproduction takes place with the help of zoospores, then it is known as asexual reproduction by zoospores. Now the next is conidia. Conidia is a very special structure. As you can see in different fungus, suppose that I am just making a fungus which is Aspergillus. So it is the Aspergillus, a fungus of class Ascomycetes. This fungus bears a special structure on its apex, which are known as conidiophore. Conidiophore. This fungus develops a special structure on the apical part, that is conidiophore, and each conidiophore gives rise numerous tiny spore-like structures which are known as conidia. So conidia are the spore-like structure which born on the conidiospores. These conidia are generally present in the form of a chain, and after dispersal, these conidia germinate and give rise to a new fungus. So in this method, as you can see, it is the hyphae. These are the conidiophore which develops on the hyphae, and each conidiophore give rise numerous spore-like structures in a form of a chain that is called as conidia, and each conidia is capable to germinate, and after germination it gives rise to a new hyphae of a fungus. Now it was the second method. Now the last one is sporangiospore. Now what is sporangiospore? I'll tell you the example of rhizopus, which is the bread mold. So suppose that it is a single hyphae of rhizopus. It is the single hyphae of a rhizopus in which the apical part is solemn and bag-like, which is known as sporangia. Inside the sporangia, the cytoplasm contracts and to form numerous spore-like structures which are known as sporangiospores. So these spores are known as sporangiospores in case of rhizopus. After liberation, as the sporangia ruptured, the spores becomes free, and after liberation, these spores germinate and to form a new mycelium of rhizopus. So this type of method of reproduction, which occurs with the help of sporangiospores, is known as a method of asexual reproduction by sporangiospore, as you can see in the rhizopus. So these are the general three ways which are found in the different fungus. These are the asexual reproduction modes: zoospores, conidia and sporangiospores. Now I will tell you about the sexual reproduction. During sexual reproduction, there are three important events takes place, in which the first one is plasmogamy, the next one is karyogamy, and the last one is meiosis. In plasmogamy, fusion of protoplasm of different cells takes place. In karyogamy, fusion of nuclei takes place, and in meiosis division, the zygote divides with the help of meiosis division and to form haploid spores. So I will tell you in detail about the different steps of sexual reproduction. Suppose that it is the hyphae of a fungus which consists of numerous cells, and these are the septa which are found between the cells. It is another hyphae which also consists of septa, and each cell bears nucleus. Now, first of all, these hyphae comes in contact with each other, and after that, their cell wall fuses and may be dissolved. So, the plasma of both the cells are mixed together and it is termed as plasmogamy. So the first step is plasmogamy, in which the plasma of two different cells of different hyphae is fused together, and the term is plasmogamy. That is number one. Now, the nuclei of both the cells comes in contact with each other. Suppose that now it is a single cell in which both the nuclei are present. These are the two nuclei. Now, after some time, these two nuclei fuse together. So the fusion of these two nuclei takes place after some time, and the fusion is known as the step number 2, that is karyogamy. Karyo means nucleus and gamy means fusion. When the nuclei, both the nuclei fuse together, then it is termed as a karyogamy process. As the result of karyogamy, a diploid zygote is formed. So it is the diploid zygote. It is 2n. Both the nuclei are n and n. So when the two haploid nuclei are fused together, then a diploid zygote is formed. So it is the diploid zygote. It was the step number 2. Now the last step is meiosis division, a division of reduction, in which the diploid zygote divides and form 4 haploid spores. So in the process of meiosis, that is the third step, a zygote which is diploid or 2n divides by meiosis division and to form 4 haploid spores, and these spores give rise to a new mycelium after germination. So it was the sexual reproduction in different kinds of fungus. So there are 3 methods or you can say 3 steps: plasmogamy, karyogamy and meiosis. Now I think everything is very clear to you. Now the last point is about the classification of fungus. So different scientists gave the classification of fungus and they proposed different types of classes of fungus. Here I told you about the 4 classes which are important according to your syllabus. So fungi can be divided into various classes. They may be number 1, Phycomycetes, and the example of Phycomycetes are Rhizopus, also known as nuker or bread mold or black mold also, and the second one is Albugo candida. Albugo is a genus and Candida is the species which is especially found in the plant of Cruciferae family, such as Raphanus sativus means reddish, Brassica campestris means mustard. So it is the parasite of different crucifers, and it is the cause of a disease which is known as a white rust of crucifer, in which white pustules appears on the back surface of the leaf. So it is due to Albugo candida. Now the next class is class number 2, that is Ascomycetes, and the important examples are Penicillium and Aspergillus. We all are very familiar with this name, Penicillium. Penicillium notatum, it's a type of Penicillium species. It's a species is helpful for the production of penicillin, which is an antibiotic, and made by Alexander Fleming in 1928, and he got the Nobel Prize for this. After that, the next class is Basidiomycetes, and the best example is Agaricus. Agaricus is the mushroom which we generally eat in our diet. So it is the mushroom in English, and in biology you can say Agaricus campestris. So Agaricus is the example of Basidiomycetes. Another example is Amanita. As I made the diagram, which is Amanita muscaria. So now I will tell you about a brief structure of Amanita mushroom. As you can see, the mycelium of the Amanita is underground and it is a saprophytic fungus. And the mycelium is tubular, branched, thread-like, and the mycelium is septate. As you can see in the diagram, the septa are present between the cells and the nucleus is present in each cell. So it is the underground mycelium. During reproduction, the mycelium gives rise a fruiting body, which is the aerial structure of this fungus, and the fruiting body is an umbrella-like structure. As you can see in the diagram, the upper surface, which is the umbrella of the fruiting body, is termed as a cap or pilus, while the lower stalk is known as stem or the stipes. Below the cap, there is a several lamelli-like structures are present which are known as gills, and inside these gills, spore formation takes place. And these spores are also known as basidiospores because it belongs to the class Basidiomycetes. So these are the spores or the basidiospores which are capable to give rise a new hyphae of Amanita after germination. So it was a simple or a brief description about the Amanita. The important thing is the fruiting body which is also present in the agaricus. Okay, agaricus, that is the mushroom. Now the last is Deuteromycetes and the examples of Alternaria and Fusaria. So these are the classes of various fungi. So it was all about the kingdom fungi, introduction, general character, and a brief introduction about the Amanita muscaria. So it was all about the kingdom fungi in this video. I hope everything is very clear to you. If you have any type of confusion, any query, then you can ask in the comment section. Thanks for watching. Have a good day.